I welcome you all in the first online refresher course in chemistry hosted by Department of Chemistry, Gujarat University, Ahmedabad. On behalf of Dr. Jagdish Joshi, Professor Director UGC HRDC, and Dr. Dilip Wasawa, Assistant Professor, Refresher Course Coordinator, Department of Chemistry, Gujarat University. Once again, I welcome you all in the next session of Mr. Pankaj Podar, Principal Scientist, Physical and Material Chemistry Division, CSIR, National Chemical Laboratory, NCL, Pune. His current research interest includes magnetic ferroelectric materials, materials for thermostics, metallic nanoparticles and quantum clusters, semiconductors including graphitic carbon-based materials, phosphor nanomaterials, biophysics. Specialty with various experimental methods such as advanced microscopic methods, which includes piezo response force microscopy, force spectroscopy, electron force microscopy, electron microscopy. Optical methods, which includes spectroscopic ellipsometry, Raman spectroscopy. Low temperature and high magnetic field setup includes vibrating sample magnetometry, squid magnetometry, AC suspectometry. Impedance spectroscopy down to 2K, magnetoimpedance, magnetoresistance, and Hall effect. Thermal method includes specific heat capacity, thermoelectric figure of merit, thermal conductivity, magneto-heat capacity. Self-assembly includes Langmuir-Blodgett method. Glimpse of some awards and achievements during his tenure. Awarded as the Fellow of Electron Microscopy Society of India in the year 2017. MRSI Medal for the year 2014 instituted by the Material Society of India. Elected as the Fellow of Maharashtra Academy of Sciences in Physical Sciences in the year 2013. Highlighted by the American Chemical Society among authors of high quality research in ACS journals in India 2012. Scientist of the Year Award in 2010 at CSIR National Chemical Laboratory instituted by NCL Research Foundation R.A. Mashelkar Endowment Fund. Discussion leader of a session on spin polarized materials at 2010 Gordon Conference on Magnetic Nanostructure held at USA in August 2010. Young Scientist Award in Physical Sciences for the year 2008 instituted by the Council of Scientific and Industrial Research CSIR India. Received Junior Research Fellowship from 1995 to 97 and Senior Research Fellowship during 1997 to 2000 from Jawaharlal Nehru University, New Delhi through its entrance examination. Under the guidance of Mr. Pankaj Podar, 11 PhD students graduated from his group in diverse fields such as biotechnology, in chemistry, in chemical sciences, in physical sciences, and in biological sciences. Currently, 4 PhD fellows are working in his group with the diverse fields. 3 postdoctoral fellows currently working in his group under the fellowship offered by DST India. Mr. Podar has 3 patents granted and more than 50 quality research papers has been published in reputed national and international journals. Thank you.
Hi, my name is Dr. Pankaj Podar. I work at CSIR National Chemical Laboratory, Pune. I'm going to speak on the title When Material Science Gets Life at Nanoscale from Microbes to Molecules. This topic is associated with green chemistry, how you can avoid harsh chemicals to make certain materials by following the laws of simple biochemistry. So this is the collage of some of the students who have worked in my group in last 15 years. And a lot of our funding comes from pharma industry mostly. So this presentation, the focus is the motto that nature is our biggest teacher. We can learn a lot from the examples which nature present around us. What we can learn from the nature is that nature employs most energy efficient processes most exotic materials, sometimes we cannot synthesize them in laboratory. Sometimes these materials can be synthesized in extreme conditions. Processes happen at extremes of the time scales, and length scale can also be found in nature. Actually, I mean, for example, you can find sudden synthesis taking place where the precursors are in micromolar concentrations in lake water. So you can still have examples where such kind of synthesis, material synthesis takes place. And we already know that energy efficiency is followed, atom economy is followed. And what I mean to say extreme conditions is basically you can say that you will get some examples very soon. So the keywords are biomimetics and bio-inspired engineering, or you can say chemical engineering or chemistry. So there are a lot of messages for chemists here. Obviously, there is a lot for biologists and biochemists. Nature has its own slow way of managing various materials on this planet, often in the extreme conditions. But rapid spread of man-made synthetic stuff in previous century might create new challenges for nature to dispose of. We can see the problem with the polymer chemistry. The polymers which are derived by hydrocarbon industries were sold off as having some kind of degradation properties for decades and decades. But it turns out that it was a lie. It's not possible to degrade those polymers so easily, and we can see that the amount of polymers are piling up on Earth and in water bodies. So how does is it possible that we learn from nature and how the processes of biomanagement takes place in the nature? So there are two ways, actually. You can see that these are the microbes, and they can interact with the ions, usually cations, and they can also interact with large macroscopic bodies. You can see the rocks. So when these microbes interact with the larger bodies, microscopic bodies, how do they manage these bodies? How do they adapt to this environment, and what we can learn, which can be useful for chemists? And similarly, when you expose them to the ionic level, how do they manage the toxicity of ions, and what can we learn as a materials chemist from those ideas? Some of the ideas came as early as 1979, and which turns out that these bacterium systems are much smarter than what we knew earlier. The interaction of cells with material science always comes full with surprises due to their sensitivity to the surrounding environment. And this is the example of magnetotactic bacteria, which is usually found in the freshwater lakes. And what you can see on your left-hand side is a transmission electron microscope image with a scale bar of 300 nanometer, and you can see that there are the synthesis of iron oxide Fe3O4, or you can call it magnetite, is taking place here. And these crystallites, nanocrystallites, are highly crystalline in nature, and they're magnetic. And the gray area, you can see this is a bacterium. So it was surprising that when Blackmore, a microbiologist, discovered that these collection of these bacteria under the microscope, they orient into certain direction, which was identified as magnetic North Pole or South Pole. So they align in certain direction. And at that time, he was not sure if there is something in the value of this microbe that was discovered later on by Richard Frankel. Richard Frankel did the transmission electron microscopy and a lot of magnetic measurements. And then he said that these are orienting because there is a presence of certain inorganic system. So you can see that this is a perfect example of organic and inorganic hybrid system. We do a lot of effort in the chemistry lab to make these hybrid systems. You can see also the self-assembly, example of self-assembly, right? You can see that particles are chaining up. So there are several questions come into mind that if you have to make this same thing synthetically in a chemistry lab, it will be quite painful. You have to maintain the ratio between ferrous and ferric ions. You have to have a particular temperature. You have to have temperature, particular pH. You have to have particular molarity, and you have to take care of the other environment, basically that co-precipitation and some other technique. You have to put NaOH to reduce the pH later on, and then you have to passivate those particles to stop their growth by oleic acid or some other surfactant. So then finally you get these particles, and that is not sure that sometimes they are not proper when you make them. Monodispersity is also an issue. So many issues are there actually. So isn't it surprising that these particles are being synthesized without those techniques, and what we can learn from that? And second part is self-assembly. In chemistry, we learn a lot of self-assembly techniques, and they're quite cumbersome. The Langmuir-Blanchet technique, solvent dried, solvent drying mediated assisted self-assembly. There are many, but they're quite complex. So here you can see they're chaining up nicely, and the distance is also quite uniform. Apart from here, there is some gap. So nature has gifted us alternative to a lot of these high energy techniques. For example, high energy ball milling, which is a mechanical technique. So biology helps materials chemistry. So I will tell you a story that we were trying to chemically synthesize BiOCl, bismuth oxychloride nanoparticles. And what we got were these large particles. They were synthesized by sol gel techniques. And you can see that what we got were like around, this scale is 2.2 micron. So you can see that each particle is quite large. So you can see that it is around 250 nanometer, and plate-like particles. But I wanted something which is 25 times less than that, around less than 10 nanometer. So these particles, which are around 250 nanometer or larger from one side, they're quite larger. You can't say that these are nanoparticles. So what do we do with those particles? So what we did, that we fed them to some fungi, Humicula. So okay, so then we fed them to these microorganisms. I don't know how to pronounce this SV anyway. So this was done in collaboration with my microbiologist friends. But idea originated in my own lab, and I asked my microbiologist friend whether his fungi will be able to reduce it. So he gave me this humicula, and then he said, okay, you can try. So one of the students in my lab, she synthesized, she basically interacted these particles with the fungi. And what you can see that the pinkish solution or suspension, so that these particles are almost settled. It's like a turbid suspension. And as a function of time, this BC is basically as a function of increasing time, they become more and more translucent. That means the size, which was much bigger earlier, is getting reduced. So you can see a lot of Rayleigh light scattering, and that's the reason that it's getting more and more transparent. So there are several processes happening here. Of course, redox chemistry is always at the play, and but primarily there is a degradation which is happening at the ionic level. Leaching out of some ions cannot be ruled out. So, for example, these particles might be fragmenting apart uh, from some of the uh, uh, weaker areas where there is a crystalline imperfections. So those areas where there is an imperfection in the crystallites might be leaching out some of these ions, and then slowly these. So this is basically mediated by some of the metal seeking enzymes basically, which go and attack on some of the cations basically. So you can see after 18 hours, this is the image we got, and you can see that particles are getting smaller and smaller, and you can see that they're fragmenting apart, and the size is here around 50 nanometer. Of course, there is a small amount of polydispersity. There is a but majority of the particles are around 15 nanometer. But if you wait long enough, you can see that the particles have finally fragmented around 5 or 6 nanometer, around 6 nanometer. So this was quite surprising that in order to reduce probably the toxicity of these larger particles, particles are getting milled down in such a small size. So there is a lot of chemistry involved. Obviously, there is, this is, microbes are using finally this biochemistry, which is quite unique. And if you notice that each particle, this scale is 1 nanometer. So what you see is the high resolution transmission electron microscopy image. You can see the lattice fringes, and you see there is a significant amount of crystallinity here. We also tried this method on hematite alpha Fe2O3 particles, quasi-spherical and nanorods, just to see whether it works. And we also changed from fungi to the yeast, and this image shows the particles, hematite particles before biomilling, and B image shows the particles after 72 hours, and here you get the particles which are after 160 hours. So this technique I named as biomilling after ball milling. Ball milling is a mechanical technique, so and this is called biomilling actually. So after that, we wanted to study the mechanical insight in this process, and then we selected another material which is of importance to pharma industries a lot, and we were already working with beta phase of iron oxyhydroxide nano spheres covered with sucrose or dextrose for intravenous injections for hyperphosphatemia. And these projects we have been doing since more than 1 decade for several pharma companies, Glenmark and Amcure, Cipla. So this is alpha phase, and this is very stable phase of iron oxide. We first used wet chemistry techniques to make these rod-shaped particles. Basically, rod-shaped particle you can easily distinguish that there is a change in the shape. So that was the motivation behind using these chemically synthesized rod-shaped particles to see the clearly where exactly the damage is happening, because earlier structures were not so much anisotropic, and it is very easy to check this process when it is you take anisotropic disease. So you can see that we started from highly rod-shaped particles, and as time progresses, you can see that there is already a lot of damage is happening. And while crystallinity is maintained, so this is uh, the dark field image, and this is bright field image. You can see that finally we got the particle size again, which is around 10 nanometer or less. So, and this is selected area electron diffraction image. You can see that typically these particles show highly polycrystalline nature. So this was also surprising that why the particle size becomes less than 10 nanometer in almost each case. So is this the particle size which is less toxic to the microbes? So there are several interesting questions which comes from the science. So you can see that these are the nanorods, and this is selected area electron diffraction image of those nanorods. And you can see that finally they are breaking apart. You can see some damages here. And after some time, you can see some more damages. You can see that these are there are some perforation, and but you can see that this is 2 nanometer scale, and you can see beautifully the lattice fringes. So crystallinity is very well maintained. After 120 hours, you can see more and more damage can be seen. And these are some, what you see on the background is a lot of protein, aggregated protein. You can see because a lot of secretion of protein happens. And further, you can see this scale is 20 nanometer. So finally, those rods are broken down into quasi-spherical particles here, and size much smaller. And this is the dark field transmission electron microscopy image, which shows that these particles are highly crystalline in nature. And this is very important image for materials chemist, because when we do materials synthesis using red chemical techniques, we know that it is very difficult to get crystalline, single crystalline material in nano dimension. So this was very motivating to us. So finally, you can see that a large area image. We also used atomic force microscopy technique to look at the mechanistic details as a function of time. You can see that these are S in size uh, alpha phase of iron oxyhydroxide nanorods. And you can see that as a function of time, you can see finally they are going from rod to quasi-spherical nanoparticles. And we did some modeling that we found that there are certain sites where they are breaking apart. And so you can see that more or less crystal structure remains maintained, but one of the plane is more prominent. You can see that this is the 0 hour. At 0 hour, you have several peaks, but so basically it is getting cut probably from one of the plane. We also applied this idea on rod-shaped zinc oxide nanoparticles. And I would like to mention here that the reason behind using this iron oxyhydroxide and zinc oxide rod-shaped nanoparticle was to prove that this method can be used to make iron and zinc fortified protein supplements, because this Saccharomyces cerevisiae is known as baker's yeast, and we put that put this same yeast to bake our bread. So this is friendly to our human system, and the protein which is inside is nutritious. So we thought that if we can have the system where we have this iron oxide and zinc oxide systems in a very small amount, and also in nanoparticle form, so this can be readily absorbed by the body for the kids who are deficient in iron and zinc, which are basic nutrients. And also you have a lot of protein naturally available, and this is a very cheap method actually. You can see that it can be scaled up very easily, and this yeast is particularly very robust. So this method, you can see that these are S-synthesized zinc oxide nanorods, and you can see that after 24 hours and 72 hours and 120 hours and 168 hours, finally you can see that these particles are getting reduced from nanorods to finally quasi-spherical particles here. Again, the same uh, results we got, that particles were finally very, very small actually. These are the AFM atomic force microscopy height images, which shows that the presence of quasi-spherical zinc oxide nanoparticles after 168 hours of biomilling. And this image shows the three-dimensional view of the same. And this shows the X-ray diffraction images from 0 hour to 168 hours to prove that there is no change in the lattice structure. So you are not making any new material, but you are fragmenting it apart. So we try to look at its biochemistry, and we did a lot of work. And for example, we put those particles inside the dialysis bag, and then we wanted to see whether cells, the physical proximity of the cells, the yeast cell is necessary. For example, we put the cells uh, outside the body. So we found that there is a potential role of protein corona secreted by these yeast cells actually. Uh, so finally, it is down to the basic uh, protein chemistry, which is very, very interesting actually. Uh, so what we found that uh, so now basic question arises, that how do these yeast cells know the presence of zinc oxide, which is inside the dialysis bag? I mean, there is no uh, physical uh, I mean, they don't feel the presence of each other. There's no interaction between direct interaction. So what? So still there is a secretion of extracellular proteins, which is cutting them down, these rods to the particles. What we found out that there is always, and it is very interesting that there is always a very subtle amount of leaching of zinc ions, which you can detect using ICP or some other high-end methods, that these zinc oxide nanoparticles or nanorods, they will always reach out. So these very minute amount of zinc ions are, since when they leach out of this dialysis bag, and they are picked up by these cells. So they are very sensitive to the environment. And as soon as they sense the presence of this zinc ions into the water, so they will start secreting these proteins, which not only try to cap these uh, rods, but will also try to break them up, chop them. So there are two functions that it will try to basically, it's like a managing the threat management, that break it and cap it immediately, right? Mask it. So this is a strategy which is used by, and many times what happens that these particles are taken inside the cellular bodies. It's not like processes are happening outside the cell, but sometimes a lot of things happen inside the cell body, which we are definitely not interested, because we don't want uh, particles to be taken inside the cell body, because it will be difficult to extract them. So I would like to now this already explained this part. I would like to explain the reverse part. So we were interested also in the reverse part, that what exactly happens once we, instead of feeding larger particles, if we feed to the microbes, the smaller, basically ions. So what exactly happens? So it is similar to what we do in the chemistry lab. So here we wanted to synthesize some form of manganese oxide, and we were not intending that we will make Mn5O8 nanocrystals, but this is what we got, and this is that makes it very interesting. So we use this fungi, Fusarium oxysporum, and these particles show the images of Mn5O8 uh, nanoparticles, and you can see again every size is around 10 nanometer, which is again surprising. In this synthesis, what we did, we fed these microbes manganese acetate, where manganese Mn is in the oxidation state +2. Now, to synthesize Mn5O8 from manganese acetate Mn+2, you need to have a lot of redox stations taking place. And it is not a simple process. Why doesn't we get MnO or some other oxides, manganese oxide? You can simply get, you can get MnO or Mn2O3 or MnO2. Why only Mn5O8? So these are very interesting questions for chemistry students, and not very easy. And another thing which you can see, that beautifully single crystalline nanoparticle, you can see this beautiful size, which is almost spherical, and you can see the lattice fringes. These are transmission electron microscope images, and you see the scale 1 nanometer. So another work was based on cobalt oxide nanocrystals, and it was again a similar case, that we use a marine bacterium this time. So this is one of the first example from our lab where we didn't use yeast or fungi. We used bacterium isolated from marine water, and what we found, that when we fed again cobalt acetate, we got not COO or CO, some other oxide. So we got CO3O4, where you need to change the oxidation state of +2 to +3 as well. So you need a proper ratio of CO2 and CO3. So how does this bacterium manages such a subtle chemical change is quite unique in its own, basically, that it's not that easy to uh, understand. And remember that everything is happening in ambient conditions. There is no high temperature, high pressure, or use of various solvents, or control, human control. Nothing is involved. I mean, there's no human control required. It is happening on its own. So this was published in Chemistry of Materials, which you know that it's a high impact factor journal. So again, what you see, that these particles are highly crystalline in nature, and we did a lot of detailed X-ray photoelectron spectroscopy analysis. And in this, this was featured in many news articles, including Nature India. Uh, at the time, there was a lot of excitement. And in this case, we wanted to know what is the effect of these cobalt ions on these bacterial cells. We have seen a lot the effect of microbial cells on either large particles or smaller ions, that we can see that what kind of materials chemistry is taking place. But we wanted to see the reverse happening, and that became another very interesting study for us. And what you see is the top, that these are the smaller, uh, these are actually basically bacterial cells. And And this is a controlled study that this is scanning electron microscopy image. And you can see that these cells are happy, isolated, they are separated, some of them are aggregated. And this column shows time-dependent controlled studies where these cells were not exposed to any cobalt acetate ions, cobalt ions. And you can see that these are well separated as a function of increase in the time. But when you, and this column shows the cells once they are exposed to the cobalt ions, and you can see clearly that here these uh, bacterial cells start getting agglomerated, and they show basically a kind of like a cooperative social behavior. And it is quite unique that in order to manage the stress, or in order to manage toxicity, which now they have experienced, so they come together. So they signal each other, which is called quorum sensing. So it's basically a chemical signal which they send out to each other. And there are two things which are happening. So they make outer sacrificial layer. So outer cells, they choose to die, but inside they survive actually, or they basically form spores also. So this is very unique, very interesting behavior, which is often seen in the biology world, in not just in bacterial cells, but you can see this behavior in ants also. They when they when you throw water, so ants will form like a ball, and ant starts floating. So in multicellular organisms also, this kind of behavior is seen. So it's quite, and also another thing is that they start secreting a lot of extracellular protein, like you can see biofilm. So, and it happens in when you expose them to antibiotic molecules also. And that is one of the reasons that many times drugs do not work on pathogens. So these ideas are also useful when you want to make some, um, this can act like a model study if you want to study the antimicrobial surfaces, for example, surgical tools or certain things. Some of these ideas can also be taken to viruses. Um, so this was the question we asked. Uh, we wanted to answer using fundamental chemistry, that does ferroelectric tetragonal phase exist in sub-10 nanometer barium titanate nanoparticles? So barium titanate in bulk shows ferroelectric properties, and it is known that from bulk to nano, the material undergoes a lot of changes in the physicochemical properties. So barium titanate nanoparticles, barium titanate bulk particles are tetragonal in its crystallinity in the bulk phase, And when you reduce the particle size, so tetragonal shape undergoes cubic uh, change. So basically, there is a change in the symmetry. So it is going from lower symmetry to higher symmetry when you reduce the particle size. So when barium titanate becomes cubic, so it loses its ferroelectricity, and so they become paraelectric. In a ferroelectric material, you can polarize the uh, lattice. So this is very useful for many applications. But ferroelectric materials are not very useful. So if you want to miniaturize the devices, some memory devices, for example, ferroelectric RAM. So this was the question that what will be the fate of barium titanate when you reduce the particle to the sub-10 nanometer dimensions. So there are lots of papers, thousands of papers, which are based on either ball milling the larger particles to make them uh, much smaller, or using chemistry methods. All these methods, they report that there is a change from tetragonal to cubic phase, and there is a limit which they call that below certain limit around 40 or 50 nanometer. They report that material becomes paraelectric. So we were asking this question that whether surface plays any role in stabilizing the ferroelectric phase. And fortunately, so this was also featured a lot in many newspapers. This is a snapshot of Telegraph. This picture of my own lab. Uh, Beshaki is a faculty member at State University in New York, Buffalo, and Umesh is a faculty member in Central University, Gujarat, Gandhinagar, and Imran was at Aligarh Muslim University. Now he's at SRM University. So here we again used a greener method where everything was happening, reaction was happening at room temperature. We took barium acetate and K2TiF6 for titanium precursor, and fungus was used as Fusarium oxycorum. And this was published in JACS in 2006. And this reaction, obviously, as you have seen that it is slow. It is not as fast as the chemistry method because biology, nature takes its own time. And you see very good crystallinity and size is always less than 10 nanometer. So there is an inherent control over size. Above all, stabilization of tetragonal ferroelectric phase, which was very important, which we proved for the first time, and that's why this was a JACS paper, which is a top chemistry paper. So what you can see here is that this is a comparison between between two different temperatures. X-ray diffraction peak around 44 to theta, and you can see that at room temperature it shows typical splitting of between 002 and 200, which is unique to tetragonal symmetry. But when you heat this material above the Curie temperature, you can see that these two peaks merge into one peak. So that shows that there is a tetragonal, so there is a ferroelectric to paraelectric phase transition. And at room temperature, this phase is tetragonal. So this was very unique. So how this green chemistry method is able to stabilize the tetragonal phase, which is not so easy by other methods. So the key lies into the surface, as I mentioned, that it was predicted in a theoretical paper using some DFT calculations that if you the crystalline phase which you would like to get is controlled a lot by surface chemistry. So here, surface is covered by all these biomolecules. So these biomolecules are somehow responsible uh, of stabilizing the bulk symmetry even in the nano size. The mechanism is very difficult to predict, but this is what is happening. And also we showed that these are atomic force microscopy images, and we use Kelvin probe microscopy, which is a tool of atomic force microscopy, where you use some bias voltage you apply actually, and then you can write negatively or positively. So we could read and write actually. We could apply plus 4 volt and minus 4 volt, and you can see that. So we can write information on these grains using atomic force microscopy. So these are it shows that this is indeed a very interesting uh, technique. Uh, so this technique has a lot of potential, as you can see. Thank you very much.
Thank you very much, sir. It was very nice and informative session for all. Thank you very much once again.